Okay, in this video we're going to talk about apoptosis, autophagy, and proteasomes, as well as some concepts in aging. Now, cells that have become obsolete or damaged need to be taken out of our system in order for them to not disrupt homeostasis or even not become cancerous. Now, one of the ways that cells can kind of clean themselves up is a process called autophagy, where you have self-eating. And it's basically a process where we can dispose of non-functioning organelles or even cytoplasmic bits of material. Now, we can do this by forming what we call an autophagosome, where these organelles and material are packaged into a vesicle, and then that's fused with lysosomes in order to degrade that material for recycling and removal. Now, in terms of proteins, unneeded proteins are actually marked for destruction by ubiquitins, and ubiquitins are just a class of molecules that can basically stick onto non-functioning and disruptive proteins or even unneeded proteins. And what happens is these ubiquitins can help those unneeded or non-functional proteins be shuttled over to the proteasome. And the proteasome is basically a series of enzymes that disassemble this ubiquitin-tagged protein, and that way those amino acids can be recycled by the cell, and ubiquitin can be recycled to go out and tag other unneeded proteins. And this is just two ways that cells keep themselves clean. Now, apoptosis is basically where the cell can have programmed cell death, or basically cellular suicide. And it causes certain cells, like cancer cells, infected cells, or old cells, to neatly self-destruct. Like, they don't just explode and spill their mitochondria or other organelles out into the extracellular fluid. The process begins when mitochondrial membranes leak different chemicals that activate these enzymes called caspases. These caspases actually cause degradation of DNA in the cytoskeleton, and that ultimately leads to a series of chemical events that causes cell death. Now, these dead cells shrink and are ultimately phagocytosed by macrophages, and these macrophages can actually help to release those cellular components back into the extracellular fluid, where other cells can recycle those biomolecules. Now, in terms of developmental aspects of cells, all cells in the body contain the same DNA. But what makes cells different in terms of their function is basically what genes are expressed. So developmentally, what this boils down to is that we need cells to differentiate into certain cell types that become certain tissues and organs. So chemical signals are one of the major factors in this process, where certain chemical signals in an embryo, like a developing organism, cause cells to develop in certain ways, where certain genes are turned on or off, and that causes cells to have specific functions. Now, this develops specific cells, and we call this process differentiation. Now, when organs are well formed and functional, we need cells to divide in order to basically grow these organs. But in adults, basically, once those organs are already well formed, we just need cell division to basically replace short-lived cells or repair wounds. And some different terms that we can use to describe how cells change in adulthood are things like hyperplasia and atrophy. Hyper meaning a lot of, plasia means growth. So hyperplasia is accelerated growth that increases cell numbers when needed. And atrophy is basically a decrease in the size of cells that results from a loss of stimulation or use. Atrophy is a term that really illustrates that adage, you use it or lose it, really well, because in our body, if you don't use a tissue, it'll atrophy and therefore decrease in size, and in that sense, you'll lose it. And so you see this in aging, where if someone stops using their bones or stops using muscle or even their brain, these tissues will atrophy, and you'll see loss of function that correlates with this disuse. Now, in terms of aging, the mechanisms of this are very unclear, but there are several different theories that can describe aging as a process. There is no one way to age. Everyone ages differently, and this is why we need multiple ways to describe aging, because it's dependent on multiple different factors. So the first one here is this wear and tear theory, where a lifetime of chemical insults can cause free radicals to damage cells that have cumulative effects on the cells that can lead to age-related cellular decline. Mitochondrial theory of aging tells us that free radicals released from mitochondria can diminish energy production, and do that due to the loss of ATP. You get less metabolism, cells can't maintain their structure and function very well, and this could actually promote age-related decline of cells and tissues and organs. Other immune system disorders could actually explain aging in the sense that autoimmune disease could damage tissues, or progressive weakening of the immune response could promote cancer and accumulation of debris in the body. We also have the genetic theory of aging, which tells us that a cessation of mitosis and cell aging can lead to damage of tissues. And telomeres are important to talk about with respect to genetic theory here, because telomeres are strings of nucleotides that protect the ends of chromosomes. They're kind of like the caps on shoestrings, where they prevent those shoestrings from being damaged. And what's interesting is that every time a cell divides, these telomeres shorten. Because due to how cells divide, DNA polymerase can't copy the last little segment of DNA, which means the copy is a little shorter than the original. Now, if you multiply this out times hundreds of copies of cells, ultimately what happens is your DNA becomes shorter and shorter and shorter. Now, to avoid like cutting into useful genes, our chromosomes have telomeres, which are basically these nucleotides at the ends that are just repeating segments of nucleotides, whose function is to basically serve as a buffer. That way you don't cut into genes every time a cell divides. Now, we do have telomerase enzymes that can lengthen telomeres. However, you find that telomerases are absent in adult cells, except for cancer cells. And what this means then is that if you divide a cell so many times, eventually you're going to cut into the gene sections of your DNA. And at that point, the cell can't survive. Once you start to lose those genes because of shortening of DNA, those cells will eventually die. Now, cancer cells avoid this process, though, because a lot of cancer cells that are vigorous actually produce their own telomerase, and telomerase extends those telomeres, and that way they can just divide indefinitely. In fact, we have cell lines, scientists do, that are basically cancer cells from 40 years ago, and these things are still dividing in labs today. But it can all be traced back to a tumor in someone back 40 years ago.